Hi students, today we will learn about seed, types of seed, germination and types of germination. Let's see each one in detail. Let's learn what is seed. Seed is an important part of a plant which give rise to a new plant. Why seed is important part of the plant? Because from the seed we get the new plant. That is why it is important part of a plant. And seed forms after fertilization. Seed forms after fertilization. Now let's see how seed is formed. In the previous video, we have already learned there are two male gametes in the pollen grain. From that, one male gamete fuses with the female gamete. And form zygote. This zygote develops into embryo. This embryo again divides into plumule and radicle of a seed. Now there is one more male gamete remaining. The other male gamete, the second male gamete fuses with, there are two polar nuclei. With these two polar nuclei, the second male gamete fuses with and then form endosperm. Once again, there are two male gametes in the pollen grain. From that, one male gamete fuses with the female gamete and form zygote. This zygote grow and develop into embryo. This embryo divides into plumule and radicle of a seed. The other male gamete fuses with the two polar nuclei and form endosperm. This endosperm nourishes this embryo. Now let's see the structure of seed. The outermost layer of the seed is called testa. T-E-S-T-A, testa. Then the inside part, this part is called cotyledon. C-O-T-Y-L-E-D-O-N. Cotyledon. Then this part, this is called plumule. And then this narrow part here down, it is called radicle. Together, embryo. Here there is a small hole through which the seed breathe in and out and absorb water. That is called micropyle. Then here it's where the seed is attached to the wall of the ovary. It is attached to the ovary wall. This part is called hilum. Okay, where the seed is attached to the wall of the ovary is called hilum. The outermost layer is called testa. Inside is cotyledon, and this part is called the plumule, and later on it turns to shoot. And then the down narrow part is called radicle. Later on it turns to root. Plumule and radicle together say embryo. Then next is there is a small hole through which the seed breathe in and out and absorb water is called micropyle. Then the place where it is attached to the ovary wall, that is called hilum. Let's see the classification of seeds. Seeds are classified into two. Number one, monocot seeds. Number two, dicot seeds. These are the two types of seeds. Monocot seeds. What is monocot seeds? Seeds have only one cotyledon. It will have only one cotyledon. One cotyledon only will be there in this kind of seeds. That is why these seeds are called monocot seeds. Then dicot seeds will have two cotyledons. Dicot seeds will have two cotyledons, whereas monocot seeds have only one cotyledon. That is why these seeds are called monocot seeds. We'll see the parts of monocot seed. The outermost layer of the seed is called testa. Then inside this part is called hard endosperm. Hard endosperm. Then inside this part is called soft endosperm. Soft endosperm. Then this part is called scutellum. Scutellum, which is modified cotyledon. Then this part is plumule. Later on it turns to shoot, and this is called a radicle, which turns to root. These are the parts of monocot seed. The outermost layer, which is hard, is called, or the cover of the seed is called a testa. Then the inside part is hard endosperm. In monocot seeds we can see the endosperm inside the seed. Then this part is called soft endosperm. There are two parts. Endosperm has hard and soft. Then comes scutellum, which is modified cotyledon. Then comes plumule and then radicle. Plumule and radicle, this is part of embryo, and this plumule turns to shoot and radicle turns to the root of the plant. This is the structure of dicot seed. Okay, now here this outermost layer is called testa. Then inside these two are cotyledons. Cotyledons. Then comes radicle. Then this is plumule. Plumule. Testa, the outermost layer. Then there are two cotyledons. Then comes radicle, which later on turns to root. Then comes plumule, which later on turns to shoot. And these cotyledons are very thick and fleshy, and they look like wings like structure they are, and these are called cotyledons. These are the parts of dicot seed. Let's see what is germination. Germination is the process by which a seed grows into a new plant. What is germination? It is the process by which a seed grows into a new plant. That is called germination. Conditions suitable for germination. For a seed to germinate, there are some conditions. They should be suitable. In suitable conditions only, a seed can germinate. What are those suitable conditions? Number one, the presence of adequate moisture. Number two, the presence of air or oxygen. Three, the suitable temperature. The presence of sunlight. These are the 4 suitable conditions for a seed to germinate. Number one, the presence of adequate moisture. Moisture should be there. Adequate moisture should be there. Then the seed will germinate. Then the presence of air or oxygen. Oxygen should be there. Then the seed will germinate. Then suitable temperature. Temperature should be suitable. It should not be too hot and too cold. The presence of some light. Some light should be there. Then only seed can germinate. These are the 4 suitable conditions for a seed to germinate. We'll see types of germination. There are 2 types of germination. Number 1, epigeal germination. Number 2, hypogeal germination. These are the 2 types of germination. We'll see one by one in detail. First we'll see what is epigeal germination. Now can you see here? This is epigeal germination. Okay. Now I want all of you to see here. This is soil. Okay. This is soil. Here you can see this is soil. Now here there is a seed which starts already germinating. Okay, this already started germinating. The seed is here in the soil. This is the first step. And in the second step you can see here a root is developed and then the seed has come out of the soil. And here this part you can see, this is seed cover. Seed cover. Then you can see here this is cotyledons. Cotyledon. Okay. Then these are the 2 new leaves or the first new leaves. Okay, these are the first two new leaves. Then here you can see this third step. These roots are also developed some more and then the shoot has gone up. Still here the cotyledon is there. This is withered cotyledon. Withered cotyledon. Okay. This is withered cotyledon. Then this is again leaves. Little more, these leaves are leaf. Okay, leaf. These leaves are little more it grew. Okay. Now here in this diagram you will see that cotyledons will be coming out of the soil. In epigeial germination, the seed cotyledon will be coming out of the soil. That is what here in this epigeial germination. In the epigeial germination, cotyledons come out of the soil. Here the soil, seed, and already started germinating, and then it has come out of the soil, new leaves has come out, and then still after growing little bit more, stem is formed, and then it's a shoot. This is root and the shoot is developed and leaves are there, and still the withered cotyledon is there. That is what in epigeial germination. In epigeial germination, usually the cotyledons will be green in color, and they provide the food for the seedling. And usually this epigeial germination we find in dicot seeds. Okay, in dicot seeds this epigeial germination will be there. Cotyledons will be coming out of the soil, and after 2 days it withers and falls off. Now let's see hypogeial germination. Here again this is soil, and here is the seed. You can see the seed is in the soil. It starts already germinating, and here the second step again the root is formed and the shoot is also formed. But here the cotyledons are below the ground or in the soil. These cotyledons are there in the soil. It has not come out. Now the third stage again you can see here the cotyledons are here still remaining. Okay, and the root is formed, shoot is also formed, leaves are there, and then this is leaf. This is stem or shoot you can see, and this is root. Cotyledon is still, okay, cotyledon is still here in the soil. Cotyledon is still in the soil, and after few days it will wither and then it will fall off, but it will never come out of the soil. And they are not in green in color, and they provide the nourishment for the or food for the plant for few days, and that is their work, these cotyledons. But they are not in green in color. This kind of germination we can find both monocot and dicot seeds. But in epigeial germination it can find only in dicot seeds. That kind of epigeial germination can be found only in dicot seeds, whereas hypogeial germination we can find both in both monocot and dicot seeds. If you like my video, please like, share and subscribe. Press the bell icon to stay tuned.